Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, so picture this, right? Like a hidden world of viruses just quietly circulating out there. And not in, the usual suspects, but in bats and shrews. We're going deep today on some brand new research that's figuring out how we can even find these tricky viruses. And, get this, what they found out about, where these viruses are popping up unexpected places. We're talking specifically Zambia here.
Yeah, it's fascinating stuff. Today we're unpacking two studies that are, well, they're pretty groundbreaking. The first one, it's all about creating a new way to detect these bat-borne and shrew-borne hantaviruses, like a whole new set of tools. The second one, it takes a close look at a region in Kazakhstan. They wanted to see how common hantavirus exposure really is there and try to pinpoint what might be putting people at risk. It's like, getting a VIP pass behind the scenes of cutting-edge virus research.
Exactly.
Yeah.
And that's what we're doing here. We're going to break down these studies, pull out the nuggets of gold, the stuff that really matters, and lay it all out for you. No scientific jargon, no getting lost in the weeds. We just want you to get it. Why this matters, what it means for us.
So let's jump right in. Study number one. It's a team effort. Researchers from Japan and Zambia working together. And what they've done is, well, they've tackled this big challenge head on, finding a better way to detect antibodies against these hantaviruses in, bats and shrews.
Right, because, okay, I think most people are familiar with the idea of like hantaviruses being spread by rodents, right? Like rats and mice. And we know some of those can be pretty serious: hemorrhagic fever with renal syndrome, hantavirus pulmonary syndrome. But bats and shrews, that's a whole different ball game.
It is. It really is. And there are so many unknowns there. I mean, do they even pose a risk to humans? We're not really sure yet.
So this study is like trying to unlock those mysteries.
Precisely. So they went about developing this new method. It's called a serological method, basically a blood test, right? But the thing is, it can detect antibodies against a whole bunch of these bat and shrew hantaviruses all at once.
Wow, that's like way more efficient than the old ways of doing things.
Oh, absolutely. And it gives us a much clearer picture of what's actually going on out there.
Okay, so I'm curious, how do they actually create this new tool, this new test? What's the science behind it?
All right, so they essentially built these lab-grown versions of specific proteins from the viruses. Think of them like unique fingerprints for each virus, the nucleocapsid protein in particular. And they zeroed in on 5 bat viruses: Bruno, Dekrong, Kazan, Robina, and Xuansong. And then 6 from shrews, Asama, Altai, Kaobang, Nova, Siwis, and Thotapalayam.
So they've got like this whole arsenal of targets, basically.
Exactly. And then they created tests that could zero in on the antibodies that our bodies would make if we were exposed to these viruses. But here's the catch. They had to make sure the tests wouldn't get confused, wouldn't accidentally flag antibodies from a different but similar virus. That's what's called cross-reactivity.
False positives, not good.
Not good at all. So they had to be really careful there.
And how they do? Did they manage to avoid those false alarms?
For the most part, yeah. They did a great job. The tests were pretty accurate. They could usually pinpoint antibodies for a specific virus without, raising a red flag for a different one. But there was a little wrinkle. In the lab, they did see some one-way cross-reactivity between a couple of pairs, Quaison and Robina, and Siwis and Kaobang. It's like having a key that fits one lock perfectly but can also turn another one, but only just a little bit.
Okay, interesting. So it wasn't perfect, but still pretty impressive. So once they had this new tool, what did they do with it? Where'd they go from there?
This is where it gets really exciting. They took their test and used it to analyze blood samples from a massive collection of bats in Zambia. We're talking 1,764 bats, all collected between 2006 and 2018.
Zambia, huh? That's not the first place that springs to mind when I think hantavirus. What did they find out?
Buckle up, because the results were not what they were expecting. A sizable chunk of those bat blood samples, a significant percentage, they actually had antibodies that reacted to the mixed antigens.
Okay, hold on. Back up for a sec. What exactly are mixed antigens?
Right, good point. So remember those fingerprint proteins we talked about earlier? They combined those from both the bat and shrew viruses, creating this cocktail of identifiers. That's what they're calling the mixed antigens.
Okay, got it. So they found antibodies that reacted to this mix. Which
means those bats had been exposed to something in that mix, but they couldn't tell at first glance exactly which virus. But here's the thing. To be specific here, 11.4% of the samples had antibodies against the bat virus proteins. And get this, 17.4% reacted to the shrew virus proteins. That's even higher.
Whoa. That's a lot of bats with potential exposure. So which viruses exactly were they finding evidence of?
Well, let's see. From the bat viruses, they found reactions to Brno, Dacron, Kazon, Swanson, and Robina. And from the shrew viruses, they saw Asama, Altai, Cowbang, and Thotapalium. Now, one interesting thing is that none of the bat samples reacted to the Nova or Siwis viruses.
Okay, so just to be clear, this is suggesting that bats in Zambia haven't just been exposed to the usual bat hantaviruses. They've also been exposed to the ones we usually associate with shrews, like those lines are blurred.
That's exactly what it suggests, and that's a pretty big deal.
It is. And you mentioned earlier that cross-reactivity, that a slight possibility of a false positive could be an issue. Did that show up at all in the actual bat samples?
Okay, so this is important, right? They specifically checked for cross-reactivity between the different virus proteins in those bat blood samples, and guess what? They found zero evidence of it. Nada. Zip.
So that basically confirms that the bats were truly exposed to multiple viruses, right?
Exactly. It wasn't just the test getting confused.
Okay, that's huge. So, big picture. What does this research, this new tool, tell us about hantaviruses in Zambia?
This new screening system, it's a game changer, honestly. It gives us this incredible power to understand these lesser-known hantaviruses, where they're spreading, how they're moving between populations. It's a big step forward in tracking these viruses.
It is. All right, so that was a deep dive into that first study. Now, let's shift gears and head over to West Kazakhstan for the second one. What were the researchers looking at there?
So this group of researchers, they wanted to know how common hantavirus exposure is among adults in this region of Kazakhstan.
Okay, and why Kazakhstan specifically? What made them focus on that region?
Well, it's a predominantly rural area, and they've had some cases of hemorrhagic fever with renal syndrome there in the past. That's a pretty serious illness caused by certain hantaviruses. So they were thinking, okay, there's definitely a risk of hantavirus infection here.
Yeah, makes sense. So they wanted to figure out how many people in these rural communities had actually been exposed to the virus. How'd they go about doing that?
So they did the study back in 2023. They had 921 adults from rural villages, all considered high risk for hantavirus because of cases in the last decade. They had the participants fill out these questionnaires about their lifestyle, and of course, they took blood samples.
Blood samples are like the gold standard for this kind of stuff, right?
They are. They really tell the story. So with the blood samples, they were looking for a specific type of antibody called immunoglobulin G, or IgG. These antibodies stick around in your blood long after you fought off an infection. They're like a little memory of past encounters with a virus.
Like a little souvenir?
Exactly. And specifically, they were looking for IgG antibodies against a few rodent-borne hantaviruses that hang out in Eurasia, Hantavirus, Dobrovavirus, and Pumalavirus.
Gotcha. So what'd they find? How many people had those antibodies?
All right, drum roll, please. 3.1% of the participants tested positive for those IgG antibodies, meaning at some point in their lives they had been exposed to a hantavirus.
3.1%. Is that high or low?
Well, it's relatively low, actually, but here's the kicker. None of those folks, not a single one, remembered ever being sick with anything that sounded like hantavirus. No hospital visits, no symptoms they could recall.
So they had the antibodies, but no memory of ever actually being sick. That's wild.
It is. It really highlights how sneaky these viruses can be. You might get infected, but you might not even realize it.
Okay, so of those who tested positive, were there any specific hantaviruses they seem to have encountered more often?
Yeah, the most common one they saw was Pumalavirus, so it seems like that's the one to watch out for in that part of Kazakhstan.
Pumalavirus. I'll make a note of that. So they had all those questionnaires about the participants' lives, right? Did they find any links between certain activities or lifestyles and a higher risk of exposure?
Yeah, they did find a couple of interesting things. They noticed a higher percentage of positive results, people with the antibodies, among office workers. That was kind of surprising, especially in a rural setting.
Office workers. I wouldn't have guessed that. Why do you think that might be?
They had a theory about that. They figured that in these rural areas, a lot of the offices are probably older buildings, maybe not as well maintained, more places for rodents to hide. And rodents, well, they're the main spreaders of these hantaviruses.
Makes sense. So even if you're working in an office, it doesn't necessarily mean you're safe from these things.
Absolutely. It's a good reminder that those risks can pop up in unexpected places.
What about living near ponds? I feel like we've heard that before with hantaviruses.
Yep, that one's a classic. They did find a link there, too. People living closer to ponds were more likely to have those antibodies. And that fits with what we know about how these viruses spread because ponds, all that vegetation, it creates the perfect environment for rodents.
Rodent paradise.
Pretty much. So more rodents, more chances for the virus to jump to humans.
Right, of course. Okay, so they found those two links. Were there any other factors they thought might be important but didn't turn out to be?
Surprisingly, they didn't find any strong connections with things like age, gender, whether people had farm animals at home, or even if they'd seen rodents around their house.
Even if they'd see rodents. Really?
Yeah, I know, right? It seems counterintuitive, but that's what the data showed. Although it's worth noting that a large percentage of people, 38% in fact, did report seeing rodents in the past year.
So rodents were definitely present, but maybe not the only piece of the puzzle. So what was the main takeaway from this research in Kazakhstan? What did they learn?
Well, even though the overall number of people with antibodies was low, it still tells us that there's a risk of hantavirus infection in that region. And the fact that nobody who tested positive remembered being sick, that suggests that the official numbers might be way too low.
Because people are getting infected without even knowing it.
Exactly. And that means the true burden of these viruses could be much higher than we realize.
Wow. Okay, so we looked at two very different studies, one in Zambia, one in Kazakhstan, both shedding light on hantaviruses in different ways. So big picture, what are the key things you think our listeners should take away from all of this?
All right, so first, that Zambia study, it really flips the script on how we think about hantaviruses. It's not just about rodents anymore. Bats and shrews are in the mix too, and they're carrying their own unique set of these viruses. And the fact that bats in Zambia have been exposed to both bat-borne and shrew-borne hantaviruses, that raises a lot of questions about how these viruses are moving around, jumping between different species. For you, this means we have to broaden our horizons and think more broadly about where these viruses might be lurking.
It's a reminder that there's so much we still don't know about the world of viruses.
Exactly. And the second study over in Kazakhstan, it's a wake-up call, really. Even in places where we know hantaviruses exist, we might be underestimating how many people are actually infected. And those unexpected findings with office workers, that highlights the fact that these risks can be hiding in plain sight, even in seemingly safe environments.
That's a sobering thought.
It is. But the good news is, the more we learn about these viruses, the better equipped we are to protect ourselves.
Absolutely. So why is it so important to keep researching these viruses? Why should we care?
It's all about public health, protecting people from getting sick. The more we understand about hantaviruses, where they are, how they spread, how they affect us, the better we can track them, prevent outbreaks, and develop treatments if needed. The development of those new detection tools, like that one in the Zambia study, that's a huge win for public health.
It's a good reminder that animal health and human health are deeply connected. What affects one often affects the other.
100%.
And it makes you wonder, right? What other viruses are out there, hiding in animals we haven't even thought to look at yet? What other surprises does the world of viruses have in store for us?
That's the big question, isn't it? And that's why research like this is so crucial. It's like shining a light into the darkness, revealing a little bit more of the puzzle each time. We're just scratching the surface.
And who knows what we'll find next?
Exactly. It's a fascinating and ever-evolving field, and for you, it reinforces the importance of staying informed, following the science, and understanding that our health is connected to the health of the planet.
Could have said it better myself. Thanks for joining us today for this deep dive.
Yeah.
We'll see you next time.
See you then.
Thanks for tuning in to the Health Headliner podcast by Todo Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.